All right, thank you very much. It's a pleasure to be here today. Try to find out where the mic is. The title of my talk is Biological Assays in Support of Manufacturing. So I thought I'd open by sharing with you the FDA definition of manufacture because it's more involved than one might think. Manufacture means, but is not limited to any or all steps in the recovery, processing, storage, labeling, packaging, or distribution of any human cell or tissue, and the screening or testing of that cell and tissue donor. So the definition not only covers producing the product, but also the testing of the product and its raw materials. For stem cell therapies, this means donor eligibility testing, final product testing, and in-processing testing at key steps are all part of the manufacturing umbrella. So bringing any product to market is fraught with risk. Things can go wrong unexpectedly, and this risk is really magnified when you're talking about stem cells. For one reason, the path to market is uncertain. There's a little more certainty coming with every new product that gets approved, but it's still, not a traditional biologic by any standpoint. There's also few, if any, platform technologies available for use across the market. The industry would like to get there. I don't think we're quite there yet. And because of this, there's no one-size-fits-all testing platform now, or any platform for that matter. It means that there's a high degree of customization needed to bring any cell therapy to market, and that equates to risk. On the manufacturing side, we develop assays to generate data to figure out whether or not our manufacturing process is altering our product, when to start these assays, and how much energy to put into assay development should be a topic of conversation from the very inception of the product. So picture yourself walking into a meeting with an interested venture capital firm or a hedge fund for that matter. You're confident going in, you think you've got a good chance of walking out of there with a letter of intent because you know your product really well, you think it has a great chance of helping thousands of people, and you're just feeling really good. So you start your meeting. Thank you very much. I'd like to tell you a little bit about our product. It was licensed from technology that was developed in the University of Stem Cell. It was developed by the world's foremost authority in this cell type. We've done extensive animal model testing on this, and I'm going to share some of those, and then you get cut off by one of the partners. It's a great sounding product. Can you tell us about your manufacturing plan? Well, they're going right for the chase, but you're ready for this question. For manufacturing, we've chosen to work with Joe's Stem Cell CMO. They've been in business for 20 years, and they just recently started work with a very similar product to ours, so we think they have the best. And then you get cut off again, this time by the senior partner in the firm. He just looks you dead in the eye and says, can you really verify for us the integrity of your product as it comes off the manufacturing line? And then you look around the room, you see all of the partners there staring at you wide-eyed, waiting for your answer. And this is the one question, of course, you didn't want to hear. So you kind of meekly say, well, you're asking about our assays. We have one assay that works reasonably well if Elvis runs it on Thursdays while standing on one foot. And then you kind of look around the room, and instantly all the partners are they're texting on their phone. You know you've lost them. You went from feeling really good about your prospects to this. Now, don't think that these conversations have to only be with interested potential funding sources. It could be with your CEO or your director of development as well. And of course, the topic can shift slightly too. It doesn't have to be about funding. It could be about, say, when your manufacturing validation is ready to get going. You never want to hold up a task on the critical path for want of an assay, especially if your CEO is expecting this activity to be completed in 2 months, when you know full well it's going to take 4 to 6 months to develop this assay, not even validate it, before it can even be used. So back to the little scenario, what happened with that venture capital firm? Well, they saw risk, and they saw no mechanism to understand it. Why? Because you had no assays. So imagine if you'd started off some assay development activity earlier on in your development path, and you had some real assays to lean on, and you could have offered them up a very different answer. Oh, so you're asking about our assays? Well, we have 2 great potency assays and also a validated way to count and determine our cell viability. So I can tell you in no uncertain terms that the product coming off the manufacturing line is going to be representative of the product that we'd produced in the past. Now that's a great answer, and great answers like that are a real value add. So I'm going to walk you through a hypothetical manufacturing scenario right now just to kind of highlight some of the ways that assays can really work for you. Starting early on in the inception of a product, we've got what I'm calling microscale manufacturing. Microscale manufacturing is, again, just this early effort, kind of tinkering even, just learning your product, learning how it's going to work. The research could even be focused on non-human sources at this time. But nevertheless, one needs at least an assay to count viability and count cells before proceeding. Then you're ready to move on. You're into a slightly bigger scale of production, laboratory scale manufacturing. Early assay development activities here could be centered around transitioning from your non-human to your human cell sources. It could be some characterization, trying to identify what cell types represent impurities relative to your mechanism of action. Regardless, you've got just enough material to do some early assay development and hopefully some animal studies to really get your program kick-started. You don't want to throw this labor-intensive, very rare material into a very costly animal study without at least knowing that your cells are viable and function reproducibly, and you need your assays to help tell you that. It's also this stage in the game where you should start to really think about your assay development plan. Do you have the skill sets in your company to develop these assays? Can your CMO do it? Or do you need to reach out to a contract research organization who can help develop these assays, support your manufacturing, and ultimately release product for you? You can never start to ask those questions too soon. So after you get all those kinks worked out, got some good animal data moving forward, you move into manufacturing scale-up. This is a very busy time in the development path. And the name of the game here, as we learned this morning at the FDA session from Mo, is its comparability. You have to be able to demonstrate that your material is unaffected by the manufacturing process and is representative of material that's been produced in the past. This becomes more important because sometime during this stage you're going to be transitioning away from your production at the lab or at the facility to very high dollar, high risk GMP manufacturing. You have to be able to tell that your material is unaffected by the manufacturing process. Now, as the name implies, scale up, you're also producing more and more material at this time. And that's good because you have all of these IND studies going on. For stability, how can one demonstrate stability of their product or pick a best formulation or cold storage solution without at least having a functional assay that you can rely on? And from a safety standpoint, how can one tell if, or even begin to model, whether or not the product is going to elicit some sort of unpredictable immune response without being able to count cells and assess their viability? Importantly, in this stage, you should also be banking reference material. Again, it's about comparability. This is material that you're going to eventually be basing your lot release on. So can you demonstrate concretely that this material is representative of the material that you use to generate all that great animal data with? You need to be able to have your assays in your back pocket to answer those questions. So it would be nice if all the assay development activities could be all shorn up by the time you finish all of your manufacturing development, but that doesn't always have to be a case. Assay development can continue early on into clinical stages. By now you're probably just wanting to pull the trigger and get rid of assays that aren't fit for purpose. You could be, putting the final touches and little tweaks on the assays that you want to move forward with, and importantly, you need to be making sure that the assays fit the intended purpose and can match the label claims and accurately support your product. There's a lot of other stuff going on in the assay development front as well. What about your sterility testing? What does that look like? Is there anything inherent in your product or its formulation that would complicate a bacteriostasis and fungistasis test? And how about mycoplasma? Can you use the 28-day USP test, or do you need to switch it up and use a PCR-based test because of some constraints in your product? Now's the time where you really need to pin down these ancillary assays as well as your go-to assays, because once you get to phase 3, sorry, the podium's falling apart. Once you get to phase 3, your assays should be in lockdown. It's very, very difficult to change an assay in phase 3 and beyond. It's not impossible, but it's challenging from both a regulatory and an economic standpoint. Say you've got a release assay that suddenly stops working, the consequences of that can be huge. You can't release product, you can't distribute product, and I don't even want to really ponder too long on what that would mean to the company or the product. So starting from the very earliest inception of the product, the assays that you use, the tools that you use to generate data have to be at the forefront of your mind and help push development of your product. This is a photograph taken of a piercing parlor in New York City. It was taken, the late 70s, early 80s. And someone in the back of the room is going to say, well, what does piercing have to do with assay development? Well, I'm going to tell you. If you can read the awning, I think it's very clever. It says ear piercing, your choice, with or without pain. Now, you can't convince me that getting your ears pierced isn't going to hurt, even just a little bit. And I think the same's true with assay development. It's going to hurt, but you can choose how much it hurts. If you start your assay development early on and approach it with an open mind, it's going to be a lot less painful than struggling with assay development at the end when you've got 10 different things going on in parallel paths and all sorts of other pressures. So start early and it will hurt less. So with the time that I've spent with you, I hope I've been able to convey a little sense of urgency to you and show you that, the earlier you start your assay development efforts, the better. It gives you more flexibility. It gives you more time to formulate your assays and put the whole picture together to make sure that you end up with the toolbox that you need to release product. Having that data in hand that's generated from your assays gives you the opportunity to change gears and respond to failures, tweak processes real time, rather than waiting 2 to 3 months for your assay to come through. It can allow you to set and manage realistic expectations and timelines. And the ability to do so is really based on the fact that you have these tools, these assays, that are generating data for you. And that data gives you the confidence to make these decisions and move your product along. All right, well, thank you very much. That's the material I have prepared for you, and I'll be happy to answer any questions from the audience in my remaining time.